In today's lab, I'm going to demonstrate for you a urinalysis test. What I have is 6 vials of simulated urine, and I'm using simulated urine for biohazard purposes. So all of these quote-unquote urine samples contain components that would be found in different individuals with different conditions. So one of them is presumably healthy, and the other 5 have some kind of condition which can be detected by this urinalysis test. What we're going to use is first a chem strip. So this is a urinalysis reagent strip test. What we can see on the side is that there are a series of colors. And if we zoom in, we can see that there are different substances that these strips test for. And so what we're going to do is we're going to dip one of these strips in each of the vials, right? And then after 1 minute, right? So if we read the directions here, it says read test pads at 1 minute, and leukocytes if there's a trace after 2 minutes. And so what I'm going to do for you is I am going to show you the dipping process, right, which is probably pretty straightforward, especially if you have something like a fish tank or testing a swimming pool or something. And then we're going to put the strip flat onto a paper towel and wait the allotted minute, and then I'm going to show you the results. Okay, so first and foremost, here is a testing strip. Okay. Very simply, we dip the strip into the simulated urine and again lay it flat, and we can see that over time these little squares are going to change color. Sometimes they'll get darker, sometimes they'll become a different color, right? And ultimately after a minute we are going to compare the colors that we see to this chart. Note on the side of the bottle the strip handle goes up, right? So if you flip the strip around, you're going to be getting false readings. Okay, and so as that's sitting there and waiting, we can do the next strip, again laying it flat on the table while the reagents can produce the required color change. Here are the 6 samples of simulated urine that we are going to be analyzing in this particular lab. I recommend looking at the physical characteristics of each of these tubes and writing some notes within the chart provided. For example, you should look for the clarity, right? So this one might be a little bit cloudier and these are all pretty clear, right? Look for the color, right? Red is not normal. This is generally indicating that there is blood in the urine, and that will be the case in this particular lab. So right now, please pause the video, jot down some notes in your chart before proceeding to the next images, which are going to show you the urinalysis test strips for each of these different urine samples. The last test that we are going to do on the simulated urine today is a test for specific gravity. Specific gravity is a measure of the concentration of all of the chemicals within your urine. So a low specific gravity means that your urine is very dilute. A high specific gravity indicates that your urine is very concentrated. So I am going to demonstrate how we use a hydrometer, which is this right here, to measure the specific gravity of the 6 samples of simulated urine. Okay, so this here is the hydrometer. If we look closely, we can see that there are a series of numbers on this float inside the hydrometer. In order to take this measurement, we are going to remove the float, put it somewhere safe. Going to take our first sample, sample A of urine, and fill the hydrometer until it's about 2/3 of the way full. Next, we are going to take our float and gently insert it to the cylinder. Note the displacement. This is why we only fill it 2/3 of the way full. And before we release the float, we give it a little bit of a spin. Okay, so a very gentle spin, and that prevents the float from sticking to the side of the cylinder and giving you an inaccurate reading. Okay, I am going to spin the hydrometer so that we can see the markings over here. Okay, note that the reading 1, right, 1.0 at the top is out of the urine. Okay, so that is 1.0, that's essentially pure water, right? And as the specific gravity, so the concentration of your urine or this particular urine sample increases, the float is going to float higher and higher. And so we see that the numbers are going to get higher and higher as well. Now, the way that we read this is by looking at the bottom of the meniscus. Right? So note that there is a curve, a meniscus. You never read the sides of the meniscus, you always read the bottom of the meniscus. And so here we can see that the bottom of the meniscus is at about, if we look up here, so 2, it's about 2, 2. Okay, so we would add that to the 1 up top, and we would get 1.022. Okay, and I will give you further examples of this on paper, but this is how you read specific gravity, and this measurement essentially indicates that sample A has a higher specific gravity than normal, so it is relatively concentrated with solutes. Okay, and so a less concentrated urine, the float would be farther down here. Okay, so less concentrated, more dilute urine, right? But as we can see, this is filled with relatively more solutes, and therefore the float is going to float even higher. Okay, so what you're about to see in this video is photographs of this right here for all of the samples for you to measure and determine the specific gravity of each of these urine samples. Okay, so now we have seen me complete one of these exercises, but I do want to talk just a teeny bit more about this hydrometer, okay, and exactly how we read it. So as we can see over here in this diagram, this first line up here is 1.000. Okay, so that is 1. We can also see that there are a series of lines here. Okay, and so each of these lines indicates an additional hundredth to be added to 1 here. Okay, so in this case, right, in this example here, we can see that the hydrometer is floating in this urine. And the bottom of the meniscus, right, so the bottom of the curve of the fluid is right at this level here. So what this is telling us is here we have 10, and then we count 1, 2, 3, 4, 5 lines, right? So this reading would be 1.015, right? So 10 and the additional 5 is 15, right? So again, this is 1 point. Here, I'll write it out for you. So it's 1.015. Okay, so that is the reading from this hydrometer. Okay, looking at our sample here, we can see the curve of the meniscus over here. So we want to read the bottom of the meniscus. Okay, so 1.010. And this particular hydrometer has lines every 2 thousandths. Okay, so 20, all right, and can't really see through the meniscus here, but we can count backwards. So 28, 26, 24, and this one here is 22. And so the reading on this, as I just said, is 1.022. Okay. To add yet another level to this, I can tell you that these hydrometers, right, our particular hydrometer has been calibrated to be measured at about 20 degrees Celsius. The lab in which I was conducting these tests was not 20 degrees Celsius. And so we actually have to adjust our values for this temperature shift. Okay, so when you look on your chart, you'll see the specific gravity. Okay, so that's where you will write or type in 1.022 for sample A. But the next row down says the adjusted specific gravity. And so we need to add 0.003 to our result here. All right, so I will give you the example. Okay, so 1.015 is the raw specific gravity, but to adjust this value for the temperature, we need to add 0.003, so 1.01, and we add 3 to 5, so that would be 8. So 1.018 is the true adjusted specific gravity for this particular sample. Okay, so your job will be to look through the next images. Okay, so B through F samples, and you are going to measure, right, analyze the specific gravity, type in the raw reading, and then add 3 to the last, right, to the last digit, right, in the thousandths place right here. Okay, make sure that you fill in all of this information and then look back at all of the results that you have, both from the testing strips and the specific gravity readings and the physical characteristics that you looked at before. And what you're pretty much going to do is look for the values that aren't exactly normal, the ones that are kind of outside the normal range, right? And so, for example, if there is high glucose, right, you shouldn't see any glucose within the urine in a healthy individual. And so glucose might indicate that the individual has diabetes, for example, okay, or some kind of condition related to diabetes. Okay, so you're looking for the irregular variables and assigning a possible diagnosis to each of the 6 individuals that we have analyzed today, based on the results you got in your readings of the strips and the specific gravity. Thanks.